DNA, or deoxyribonucleic acid, is the hereditary material in humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. DNA replication is the process by which the genome's DNA is copied in cells. Before a cell divides, it must first copy, or replicate, its entire genome so that each resulting daughter cell ends up with its own complete genome. The basic building block of nucleic acids, DNA, and RNA, is the nucleotide, which has 3 components, which are a nitrogenous base, a sugar, 1 or more phosphates. The nitrogenous base is a purine, which includes adenine and guanine, or pyrimidine ring, which includes thymine and cytosine. The base is linked to the 1' carbon on a pentose sugar by a glycosidic bond from the nitrogen number 1 of pyrimidines, or the nitrogen number 9 of purines. The pentose sugar linked to a nitrogenous base is called a nucleoside. The sugar can be linked by its 5' or 3' carbon to a phosphate group. A nucleoside linked to a phosphate at the 5' carbon is a nucleotide. A polynucleotide is a long chain of nucleotides. The backbone of the polynucleotide chain consists of an alternating series of pentose sugar and phosphate residues. The chain is formed by linking the 5' carbon of one pentose ring to the 3' carbon of the next pentose ring via a phosphate group. Thus, the sugar phosphate backbone is said to consist of 5' 3' phosphodiester linkages. Specifically, the 3' carbon of one pentose is bonded to one oxygen of the phosphate, whereas the 5' carbon of the other pentose is bonded to the opposite oxygen of the phosphate. The nitrogenous bases stick out from the backbone. Each nucleic acid contains 4 types of nitrogenous bases. The same 2 purines, adenine and guanine, are present in both DNA and RNA. The 2 pyrimidines in DNA are cytosine and thymine in RNA. Uracil is found instead of thymine. The only structural difference between uracil and thymine is the presence of a methyl group. The terminal nucleotide at one end of the chain has a free 5' phosphate group, whereas the terminal nucleotide at the other end has a free 3' hydroxyl group. It is conventional to write nucleic acid sequences in the 5' to 3' direction. That is, from the 5' terminus at the left to the 3' terminus at the right. Moving forward, to ensure the fidelity of genetic information, it is crucial that DNA is reproduced accurately. The 2 polynucleotide strands are joined only by hydrogen bonds. Thymine and adenine has 2 hydrogen bonds, but guanine and cytosine have 3 hydrogen bonds. So, they are able to separate without the breakage of covalent bonds. Moving forward, the specificity of base pairing suggests that both of the separated parental strands could act as template strands for the synthesis of complementary daughter strands. A new daughter strand is assembled from each parental strand. The sequence of the daughter strand is determined by the parental strand. An adenine in the parental strand causes a thymine to be placed. In the daughter strand, a parental guanine directs incorporation of a daughter cytosine, and so on. Moving forward, an unreplicated parental duplex with the original 2 parental strands. The lower part shows the 2 daughter duplexes produced by complementary base pairing. Each of the daughter duplexes is identical in sequence to the original parent duplex, containing 1 parental strand and 1 newly synthesized strand. The structure of DNA carries the information needed for its own replication. The consequences of this mode of replication, called semiconservative replication, the parental duplex is replicated to form 2 daughter duplexes, each of which consists of 1 parental strand and 1 newly synthesized daughter strand. The unit conserved from one generation to the next is one of the 2 individual strands comprising the parental duplex. Let's understand semi-conservative replication through a model. If the parental DNA carries a heavy density label because the organism has been grown in a medium containing a suitable isotope nitrogen, its strands can be distinguished from those that are synthesized when the organism is transferred to a medium containing light isotopes. The parental DNA is a duplex of 2 heavy strands, shown by red. After 1 generation of growth in a light medium, the duplex DNA is hybrid in density. It consists of 1 heavy parental strand, which is red, and 1 light daughter strand, which is blue. After a 2nd generation, the 2 strands of each hybrid duplex have separated. Each strand gains a light partner, so that now 1/2 of the duplex DNA remains hybrid, and the other half is entirely light. Both light strands are blue. In this model, the individual strands of these duplexes are entirely heavy or entirely light, but never some combination of heavy and light. This pattern was confirmed experimentally by Matthew Meselson and Franklin Stahl in 1958. Meselson and Stahl followed the semi-conservative replication of DNA through 3 generations of growth of E. coli. When DNA was extracted from bacteria and separated in a density gradient by centrifugation, the DNA formed bands corresponding to its density: heavy for parental, hybrid for the 1st generation, and half hybrid and half light in the 2nd generation. Moving forward, the theory that DNA replication was semi-conservative was confirmed by the Meselson-Stahl experiment in 1958. Prior to this experiment, 3 hypotheses had been proposed for the method of replication of DNA. Let's discuss them and compare them to have better understanding of semi-conservative replication. In conservative model, an entirely new molecule is synthesized from a DNA template which remains unaltered. In semi-conservative model, Each new molecule consists of one newly synthesized strand and one template strand in dispersive model. New molecules are made of segments of new and old DNA. So, this was all. Thank you for learning. Join us again.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.